Welcome to the three-dimensional structure of proteins lecture. In this chapter, we're going to review the levels of protein structure and explain the significant features of each level. Additionally, we're going to look at and compare the functions of myoglobin and hemoglobin to extremely well-studied oxygen-carrying proteins. This overview should give you the vocabulary and insight needed to understand how it is proteins do what they do. While I won't cover it in this lecture, go ahead and read up on hydrophobic interactions in your book. Later, when we examine lipids, understanding hydrophobic interactions should help your understanding with how lipids function. You will know you are finished with Chapter 4 when you can do the following without your notes or book. Be able to define and describe the levels of protein structure: primary, secondary, tertiary, quaternary. Compare and contrast hemoglobin to myoglobin. My apologies, but we must visit the lowest level of learning, memorization. It's likely you've come across these terms in previous science courses, but it is important to know them. These terms tend to come up over and over again in biochemistry due to the focus on proteins. It is strongly suggested that if you have not memorized these terms to the point of intuitiveness, you go ahead and do so. So briefly, conformation. The fold or structure can refer to local conformation or overall conformation. Native conformation, the fold of a protein with biological activity. Primary structure, the sequence of amino acids, no intrinsic fold. Secondary structure, the folds the linear peptide takes, alpha helices, beta sheets. Tertiary structure, the folds the alpha helices and beta sheets of a peptide adopt as a whole. Quaternary structure, comprised of multiple peptide chains associating to form one protein, also referred to as subunits. Domains, specific clusters of secondary structure elements in tertiary configuration that remain folded correctly even when separated from the rest of the protein. Prosthetic group, protein elements covalently joined to the protein that are not amino acids. Before continuing, let's look at the primary structure of a protein. This is just the sequence of amino acids as directed by the genetic code after any post-translational modifications. While at this time we cannot truly anticipate the final fold a protein will take after translation, the primary sequence does give us empirical evidence as to what secondary structures are likely to appear. Sickle cell anemia is the classic example, where a single amino acid mutation results in a dramatic new behavior for the protein hemoglobin. With the sickle mutation, the protein changes ever so slightly, actually resulting in available hydrophobic interactions. That results in the hemoglobin precipitating out of solution. Since the primary structure has such a profound effect on higher orders of structure, it is important for us to know the amino acid sequences of proteins that we are investigating. Molecular techniques such as site-directed mutagenesis allow us to specifically change the primary structure and see folding consequences. Alright, considering the idea that generally speaking peptide bonds will not rotate, that does leave two other bonds that are capable of high degrees of rotation. Not including any bonds involving side chains, the NC bond following the peptide bond, be sure to review amino acid structure, is termed phi, and the CC bond following the phi bond is termed psi. These two bonds are capable of high degrees of rotation. The phi-psi bonds are used on a Ramachandran plot to determine the degree of correctness of the protein fold, while other bond angles can occur outside Ramachandran ideals, and even should be expected. Those angles are likely to impart strain to the protein. This may be an important feature of the protein. Remember, ideal really means without strain. Strain really means higher energy. So long as the energy overall is negative, the protein will fold even if some of the angles are high energy. The two most common secondary structures are the alpha helix and the beta sheet. Let's look at the alpha helix first. What we have is a cylinder of amino acids, hence helix, in which the side chains are pointed away from the line of the helix. The helix itself is formed by backbone atoms, so while the types of residues seem to indicate whether or not a helix is present, the side chains of those residues, remember the R groups, are not actually involved in the bonding pattern that makes the helix. The helix is formed by the CO group starting from the amino end or the N-terminus that is hydrogen bonded to the NH group that is 4 amino acids away. This allows these hydrogen bonds to be arranged linearly and are therefore at maximal strength for a hydrogen bond. A complete turn is 3.6 residues, so the hydrogen bonds themselves do not form a line but are slightly skewed, and the pinch, which is the length of the turn, starting from the residue measured to the hydrogen bound residue, is 5.4 angstroms. Alpha helices are very strong and compact structures, however they can be disrupted. A proline residue, remember proline side chain, cannot interact in a hydrogen bond. Residues with large side chains can cause clashing even though the side chains themselves are not in the hydrogen bonding pattern. Even side chains with charges, for example an aspartate near an aspartate, can cause charge repulsion and disrupt the chain. Beta sheet secondary structures are quite different from alpha helical structures, but they do share a few similarities. Unlike alpha helices, which are compact, tight structures, beta sheets represent an extended form of the peptide chain. Whereas alpha helices are made from a single peptide coiling back on itself, a beta sheet can involve the same peptide chain or two different chains. These are called intrastrand and interstrand, respectively. As with the alpha helix, the hydrogen bond pattern is not created by side chains. It is formed by backbone atoms instead. Again, the actual residues can be indicative of a beta sheet, but the side chains of the residues do not form the bond network, that is the backbone. Additionally, the beta sheet can be parallel, in which the two sides run in the same peptide direction, or anti-parallel, in which case the two sides of the beta sheet are formed by different peptide strands or are running in opposite directions. Essentially, anti-parallel is about opposite directions. Beginning to bridge the gap between secondary and tertiary structures are the super-secondary structures. These are specific arrangements of classic secondary structures, alpha helices and beta sheets. The common beta barrel is shown in figure 4.9 in your book. As the name suggests, it is a large hollow cylinder formed by beta sheets, sometimes supported by alpha helices. The structures shown here are less dramatic, but also common in protein structures. The beta, alpha, beta, where an alpha helix acts as a linker for two parallel, as opposed to anti-parallel, beta strands. The helix turn helix, where an unstructured span of amino acids, sometimes containing a proline or maybe just a random coil, allows a sharp turn between two alpha helices so that the helices are anti-parallel. The beta meander, or simple anti-parallel beta system, and the Greek key, which is a complex anti-parallel beta system. Note that the linkages between the beta sheets, helices, etc., shown in yellow on this figure, are all unstructured amino acid spans. These can take the name random coil, referring to their lack of overall organization. The overall 3D arrangement of a protein, including alpha helices, beta sheets, side chains, prosthetic groups, etc., is termed its tertiary structure. Note that the level of structure is only concerned with one peptide. If multiple peptide chains are involved, that will be a quaternary structure comprised of tertiary structures of each individual peptide. Take a look at collagen, figure 4.10. The triple helix, for an example of quaternary structure comprised of secondary structures formed from 3 polypeptide chains. Although some people would say that collagen is really just a high-order secondary structure. Still, it uses subunits. That is definitive of quaternary structure. Shown here are a sampling of the forces that are involved in tertiary structures. Compare this to the forces that make the secondary structures of the alpha helix and beta sheet. That's only the hydrogen bond. In the tertiary level, we have arrangements of helices and sheets, but now the side chains can also come into play, as can the backbone further. We can also see metal ion coordinations, for example, a zinc cation, which is +2, interacting with two carboxylate side chains, which would be -1 each. There can be hydrophobic interactions with nonpolar side chains. Can you name a few nonpolar amino acids? Disulfide bonds will play a very important role. Which amino acid can make disulfide bonds? Hydrogen bonds between side chains and backbone atoms and side chain to backbone atoms can appear as well, and straight up electrostatic interactions between charged species or regions. Unlike secondary structures, tertiary structures are essentially impossible to predict without any information. In other words, just looking at a primary sequence without other information will not impart anything towards the tertiary structure. Compare this to the knowledge that we can use primary structure to make reasonable guesses about secondary structure elements. Quaternary structures refer to a level of organization that use multiple polypeptide chains that come together to form a finished protein. These chains are also known as subunits. Proteins can be comprised of 2, 3, or 4 or more peptide chains if they're quaternary structure, thus referred to as dimers, trimers, tetramers. Each member of a subunit-based protein is called a monomer. So a dimer is made up of 2 monomers. These may or may not be identical peptide chains. Consider that collagen, already mentioned, is a trimer made up of 3 identical monomers. Alternatively, consider the proteasome, which is made up of, well, many subunits. A general term is oligomer, which just means several different subunits, typically identical monomers, but not necessarily. This is a more general term used to mean a chain of monomers. Another term you're going to see frequently is allosteric. This term is used in a few different ways, but means roughly the same thing. A change at one site of the protein causes differences at another site. We'll see this in enzyme control as well as protein cooperativity. As an aside, the term allosteric site used in enzymatics means a site away from the catalytic site that causes the catalytic site to change behavior when the allosteric site is occupied, typically by a regulatory element. So here we are through the major chunk of this lecture. At this point, you should be able to discuss the various levels of protein structure, especially be able to compare and contrast them. Remember, the more you can discuss these, and the more you can do so without your notes and without just reciting statements, the more you understand. By understanding topics rather than reciting topics, you really do learn the material better. Up next, we're going to review the behavior of 2 vital proteins as we prepare for our foray into enzymatics. Get used to the idea of looking at curves and thinking about how proteins need to work within their various environments. Don't worry, I'm here to help. Both myoglobin and hemoglobin are oxygen-binding proteins. They both contain heme prosthetic groups. Myoglobin has 1, and hemoglobin has 1 per chain. Myoglobin is a monomer, and hemoglobin is a tetramer. In hemoglobin, the monomers are in pairs. 2 are named alpha, and 2 are named beta. This has nothing to do with alpha helices and beta sheets. So you could describe hemoglobin as a tetramer made up of a pair of dimers, and each dimer containing 2 identical monomers, just to give you practice with those terms. The key to understanding these 2 proteins is to know what they do for the body. Myoglobin binds a single oxygen and is found primarily in muscle cells and is used for storage of oxygen. Hemoglobin can bind up to 4 atoms of oxygen, is primarily found in the blood, where it is used to transport oxygen to the periphery, i.e. away from the lungs. The heme prosthetic group is a protoporphyrin IX ring with iron bound. This iron is capable of binding oxygen. Hemoglobin can bind carbon dioxide, but does not do so at the binding site for oxygen. However, carbon monoxide is a very strong competitor for oxygen at the oxygen binding site. What's important to realize about hemoglobin in terms of binding oxygen is that oxygen binding is cooperative. This means while the first oxygen to bind may be difficult, Once the first binds, the second binds easier. Once the second binds, the third binds more easily. It is unlikely for hemoglobin to pick up all four oxygen just due to the nature of the protein and the blood system. However, presumably, the binding of the third oxygen would make binding the fourth easier. This is an example of positive cooperativity. The alternative would be negative cooperativity, where initial binding reduces the ability of subsequent binding events. Alright, let's consider how myoglobin and hemoglobin differ in the binding of oxygen. We know the following. Myoglobin binds 1 oxygen and hemoglobin binds up to 4. Myoglobin is for muscle storage of oxygen and hemoglobin is for blood transport of oxygen. Now looking at this table, we see that percent saturation of hemoglobin, the yellow to violet lines, changes based on partial pressure of oxygen and on pH. Notice the difference between venous, low pressure, and arterial, high pressure. So the question we should ask upon making this observation is, can we explain this observation in terms of physiology so that the behavior of the proteins makes sense? Yes, we can. So let's try it. Consider two sites of interest, central and peripheral tissues. Central tissues, we're talking about lungs and arteries. It is entirely reasonable to believe that oxygen levels are quite high in these areas, and the pH should be relatively high as well. Keep in mind this is comparative statements, as we're comparing central to peripheral. In the periphery, we're talking about muscle tissues here, we would expect oxygen levels to be lower. You may not be sure why, but for now, keep in mind oxygen is being converted to water by metabolism. Additionally, the pH is going to be lower. While the pH in the blood is maintained at about 7.4, remember compensatory respiratory alkalosis from chapter on acids and bases? The tissues themselves are going to be a bit lower, possibly due to, once again, metabolic processes that result in the release of proton. So notice how hemoglobin behaves. It takes a substantial amount of oxygen to saturate hemoglobin, even to the 50% mark. 50% is not too hard if pH is 7.4, about 20 or so millimeters mercury. But to saturate hemoglobin takes closer to 70 or 80 millimeters of mercury at pH 7.4. So we can see that arterial blood, which has a lot of oxygen, at least in the non-cardiopulmonary system, and is pH 7.4, is a great site for binding oxygen to hemoglobin. As hemoglobin is transferred through arteries to arterioles to capillaries, the partial pressures of oxygen are going to reduce because oxygen is being consumed in these regions. So hemoglobin is going to have reduced affinity for oxygen. Additionally, as we move to peripheral tissues, the pH is going to start dropping because of local metabolic events, especially in the muscle, which will further reduce hemoglobin's affinity for oxygen. So as hemoglobin travels to peripheral tissues, it is more and more likely to release oxygen to those tissues that need it. What about myoglobin? Myoglobin, as we can see, is very different. It doesn't care about pH, and it has really high affinity for oxygen compared to hemoglobin. The curve is hyperbolic as opposed to the sigmoidal curve of hemoglobin, which shows hemoglobin's cooperativity for binding oxygen. Myoglobin only needs a very small amount of oxygen to become saturated, and as compared to hemoglobin, and pH does not appear to have an effect. So we can presume that as hemoglobin, the transport molecule, is losing oxygen in the region of the skeletal muscles, myoglobin, which has very high affinity for oxygen and is the storage molecule, is readily capable of binding to the oxygen. This creates an excellent system for passing oxygen from hemoglobin to myoglobin and making sure oxygen is delivered where it is needed most. As you will see, this will be regions of low oxygen partial pressures and lower pH, or where metabolism is active. Please see your book for further details and try Apply Your Knowledge 4.1. To help summarize what we just covered, let's create a simplified pathway concept. In the central region, arterial, where oxygen is high and pH is normal, 7.4, we know the hemoglobin has high affinity for oxygen. For illustration purposes only, let's assume myoglobin is absent. As hemoglobin moves towards the periphery, the conditions change, resulting in loss of binding affinity of hemoglobin for oxygen. Thus, the oxygen is becoming released in and around the capillary beds. If oxygen was just released and did nothing else, we might expect only a certain amount of hemoglobin to lose its oxygen, and the release of oxygen would increase local partial pressure of oxygen. Keep in mind, for illustration in this point, the oxygen is only being released and is therefore accumulating. However, in the peripheral, myoglobin has much stronger affinity for oxygen than hemoglobin. So as the reaction is resulting in release of oxygen due to loss of affinity by hemoglobin in the periphery, the much stronger affinity of myoglobin allows this protein to bind and pull the reaction to what we see in the muscle cell. Saturation of myoglobin and loss of oxygen on hemoglobin. This exchange is what drives partial pressure of oxygen down and allows hemoglobin to keep releasing oxygen. So in our illustration pathway, the binding of oxygen to myoglobin assists in driving the release of oxygen from hemoglobin due to the reaction shift to our muscle cell products. Study this, understand it, be able to describe it. Metabolism will be much, much easier if you can. So there you have it. We have now covered everything. At this point, you should be working on being able to thoroughly describe the different levels of protein structure and compare and contrast alpha helices and beta sheets. As well, we have now covered the differences between myoglobin and hemoglobin and how the two proteins function. Make sure you can do more than just recite this information. You want to be able to, without your notes or book, explain intuitively to your classmates this information. Being able to do so will greatly assist you in learning this material. Remember, understanding comes from discussion, not memorization. For your recommended questions, I do recommend quite a few of them. Keep working on them in preparation for your workdays. I recommend all of the questions of 4.1, 4.2, 4.3, 4.4, and in 4.5 I recommend 26, 28, 29, 30, 32, 34, 35, 41, 43, 45. There you have it. Good luck in your studies, and I'll see you at the next lecture.